Yeah, Sarah is the recipient of this year's Senior Scholar Scientist Award. And for those of you who don't know, yeah, we can do a round of applause. Yay. Um, I feel especially privileged because I get to talk about it. We designed this award this year, and it's exciting that Sarah's the inaugural awardee, to recognize a graduating senior who has really exemplified what we mean by having the core qualities of a research student, of a quality research student who's been here at ASDRP for a while. And Sarah has been at ASDRP since her freshman year. Right? That's crazy. Since 2019. I'll let Rob say a few things in a few minutes about crashing the server cluster too many times. But I'll just give a brief introduction. Sarah goes to Los Altos High School, which is located in Los Altos, California, no surprise. And when she first joined the group, I think Sarah was probably one of the most timid and awkward freshmen I've ever met. And kind of hung out with a bunch of older students who began to show her around on how to do chemistry. And soon thereafter, about at the end of her freshman year, Sarah co-authored her first paper in the group, where we reported strain-specific antimicrobial activity of this natural product called Berberine against various strains of bacteria, and compared that against some of the commercially available antibiotics out there. And from there on out, we proceeded to publish not one, not two, not three, but like 5 berberine papers about this compound that is isolated from nature that elicits its biological activity only upon irradiation. And about a year into Sarah's time at ASDRP, I had this crazy idea that we could track singlet oxygen production of berberine in an NMR tube. And some of these students were crazy enough to follow along with that idea. And I think Sarah remembers those days pre-pandemic, sitting in front of the whiteboard asking, what's singlet oxygen? About a year later, we had our paper come out about 4+2 cycloadditions with singlet oxygen. And not only a paper, but also an application note with Analysis Corporation, which is our collaborator in Canada on Benchtop NMR. After getting bored of working on berberine, Sarah decided she wanted to do some computational work. And actually during the pandemic, this is before Sahar's time at ASDRP. At the beginning of the pandemic, we were bored at home and we were looking for things to do. And we said, gee, there's this new virus around. How about we do some drug discovery for it? And so that's what we exactly did. And in November of 2020, out came the first paper on using high throughput virtual screening and reactivity-informed design to study the properties of over 200 small molecules that we had designed from on the back of a napkin against SARS-CoV-2. And by the way, that is published in November. That was submitted in June of 2020, just basically 3 months after the pandemic hit the United States, 4 months maybe. And that's largely due to both the resilience of students and also due to the vast patience of Mr. Downing here while people repeatedly crashed the server. And from then on, after we got bored of COVID, we were looking for new compounds to work on. We found this fluorinated anti-cancer compound that was also an anti-COVID compound. And we proceeded to study that for a while. And so we published a high throughput virtual screen there as well. And our most recent work in using NMR spectroscopy, specifically fluorine NMR, to look at these sorts of molecules and how to optimize their reactions with fluorine NMR, was recently accepted in the Canadian Journal of Chemistry. After getting bored of that once again, Sarah decided to take on a new challenge and moved from chemical biology into straight hardcore physical organic chemistry, where she worked on our most recent flagship paper, which is out on ChemArchive now, using that same technique, fluorine NMR, to study the reaction pathway of a very mysterious multi-component cyclocondensation. And that is currently now in review, and hopefully we'll see it published in the next few months. And then Sarah got bored of that too. So now she's currently working on hetero 4+2 cycloadditions, and we're going to try to shorten the synthesis of forskolin from 22 steps, which currently is, to 5. And so without further ado, I'll let Rob say a few things because I promised him he'd get to say a few things.
Well, first, I just want to point out that I'm a bit skeptical about Sarah getting bored. I think Sarah just needs to find new ways to challenge me on her excessive use of servers and is inventing new and wondrous attacks that she performs under stealth mode, and which takes me hours to figure out and correct. But for all of you who are listening who think that sounds like fun, it does wind up usually in a one-to-one session with me, and then lots of job output getting canceled, which is never good. But I will say that given the award, Sarah is the kind of student researcher that on a daily basis, I think challenges all of us to be better, excuse me, be better than what we are. Not only student researchers, but advisors. And that's exactly what we want to produce here at ASDRP. And so for those of you that are new, or for those of you that are taking a little bit longer to come up to speed, if you want to look to a model of what it means to be a student researcher in Excel, pay special attention to the next. How long are you going to take, Sarah? 2 hours?
Not too long, because Sahar is going to lob my head off next time we have a board meeting.
So all of that being said, Sarah, thank you very much.
And I'll just close off with this before we let Sarah begin. You know, a lot to show for that sort of research. 8 published papers with 4 more in progress, including some of our earlier Karmafor work that we have yet to publish. I don't know how many conference proceedings, a TED Talk, an invited seminar at Valid NMR, 3 proceedings at the American Chemical Society National Meeting. Just a stellar student, and if you all want to see what throughput looks like, I don't know how one single student has been so productive. But okay, so, unless
Yes.
Sahar wants to say a few things about
Yeah, congrats, Sarah. I mean, I'm already feeling jealous of what Edward having in his hand, but not to mention, I have amazing team of seniors. I've joined year and a half ago, and I am amazed by how hardworking some of you are, and you're definitely a true example of it. We're so honored that we have you at ASDRP. It's just like, well done. Like, I can't be proud of you. Well done. So I hope the number of slides are not exceeding 50, because Edward is going to be in trouble. Okay.
She's quickly deleting the chapter.
I have 83 slides. Edward told me add more too, so.
Oh.
He's gonna get through all of them, and we're gonna have enough time for Sahar's group to have a presentation.
Yeah, for every slide that you go more than 20, Edward is going to get a negative point. So.
Well, Sarah talks fast, so I have high confidence. 2 slides per minute, maybe 4. So Sarah, it is all yours.
All right. Let me just share my screen really quickly. It says that I can't have my microphone on while I screen share. Can you screen share and then I'll talk?
You gotta be kidding me. Okay.
Sus.
Where did you send this to me?
I sent it to you on Slack. Wait, let me just Slack it to you again.
Okay. Too many tabs open.
We've seen that error before.
Yeah, it's because my Zoom doesn't work today, so I had to use the web browser. Sorry.
That's a Windows permission thing. We can fix that.
All right.
So Sarah, just tell me next slide when you want me to advance.
Okay, cool. All right, thank you everybody for coming. I know the slide hasn't loaded yet, but yeah, okay, great. So today I'll just be talking about the synthetic approach using Benchtop NMR, specifically non-canonical applications of Benchtop NMR towards developing anti-cancer and antiviral agents. And I also do a lot of work with natural products. So I'll also specifically be talking about the conjunction of natural products with Benchtop NMR and how we can utilize Benchtop NMR as a learning asset and towards developing, all of these really cool antiproliferative and antiviral agents. So next slide. So the first approach we're going to be talking about is basically this project that Edward mentioned earlier, the Berberine project. And so essentially this is the semi-synthesis and structure-activity relationship of novel berberine analogs as DNA-binding singlet oxygen photosensitizers. And so as you can see here, there are many natural products with medicinal potential. We'll be focusing specifically on berberine, which is a natural product coming from the plant Berberis vulgaris, which you can see at the bottom. Next slide. Oh, next. Oh, yeah. And so it's derived from L-tyrosine, and it's a natural product. So it comes in nature, and it's actually been utilized in ancient Chinese medicine for thousands of years. And so the putative mechanism of action is that it's a DNA-binding photosensitizer. Essentially, it means that when it's irradiated and it complexes with DNA, it's able to generate singlet oxygen. So it photosensitizes triplet oxygen to singlet oxygen, and that singlet oxygen is able to do guanine-specific oxidation. And so by turning guanine into oxoguanine, it's able to induce cell death. Next, okay. And so essentially in our study, this is the study Edward mentioned earlier where we used benchtop NMR to trap singlet oxygen in an NMR tube. So our first study, we compared berberine to two commercially available photosensitizers, and those are rose bengal and methylene blue. Next slide. And so here, the first thing we did was we used in silico methods. So this is what we did. We did something called TDDFT, or where you basically look at the energy states of the different orbitals. And so you can see here, we have berberine, we have rose bengal, and we also have different analogs of berberine, as well as methylene blue. And so what we measured is we measured the energy gap between the triplet excited state and the singlet ground state of each of the analogs. And we found that all of them were greater than 0.9 AeV, which is what's sufficient for singlet oxygen generation. And so to bolster our DFT work, we also wanted to go in lab. And so what we did here, this is just the experimental design of all the work with NMRs. Essentially, we had all our compounds in an NMR tube. So that was our photosensitizer. We had our oxygen obviously flushed in the headspace. We had our singlet oxygen trap, which was alpha-terpinine. And then we also had an internal standard, which is basically what allows you to measure the amount of a substance in an NMR tube, which was dimethyl terephthalate. And so by taking an NMR of that and using all of this, we basically were able to trap the singlet oxygen generated by our photosensitizers. So you can see here the alpha-terpinine, it undergoes a 4+2 Diels-Alder-like reaction, and that generates this compound called ascaridole. And so you can see those two oxygens, that's called the endoperoxide. Basically, we were able to monitor the generation of this endoperoxide over time and the NMR spectra. So if you look at the next slide. Yeah, so here you can see like the different NMR spectra over time. And so we did this with both LED and UV irradiation. And as you can see here, it's at around 6.44 ppm. You see that little doublet? Essentially what that is that's the generation of the endoperoxide. So by taking integrals of that and measuring that against our internal standard, we were able to figure out how much endoperoxide there was for every time point. So we did this over 5 hours. And so if you go to the next slide. You can basically see how we were able to analyze the different integrations of all these different peaks, and we were able to use that to derive concentrations of each of the reactants within the NMR tube for the entire 5 hours. And then by graphing that, as you can see on the next slide, we were able to have a basic understanding of singlet oxygen formation as a function of time. So here you can see we actually measured two products. One was cymene and one was ascaridole. Ascaridole is the endoperoxide product I mentioned earlier, and cymene is just a minor oxidative product. And so by measuring the concentrations of both those during as a function of time and then adding those together, we were basically able to monitor singlet oxygen formation as a whole as a function of time. And so here you can see that rose Bengal had the greatest singlet oxygen formation as a function of time, with methylene blue being comparable in the UV trials. In the LED trials, it was interesting that berberine and methylene blue performed pretty similarly. So this corroborated our hypothesis that, you know, berberine is a photosensitizer, but it also shows that berberine has room for improvement as a singlet oxygen photosensitizer, and we could utilize this function to create more effective analogs of berberine. So next slide. So here we actually synthesized two reduced berberine analogs. They're both with sodium borohydride. It's just that the reduction to dihydroberberine is incredibly fast, so it's instantaneous basically. But if you let it stir for a while, it will turn into canidine, otherwise known as tetrahydroberberine. This is basically the even more reduced form. And so by synthesizing these three analogs, we wanted to assay the effect of the C-ring reduction on its photosensitizing properties. Next slide. Another thing we did was we did regioselective demethylations of the berberine scaffolds. So you see that methoxy group at carbon 9. We essentially were able to reduce that down to a hydroxy. And so the reason that it has the hydroxy at C9 and not C10 is because of the resonance. Essentially, it stabilizes the hydroxy at carbon 9 rather than carbon 10. And therefore, we were able to just synthesize this form of berberubine, which is what it's called when it's reduced at C9. And we also wanted to see how that would affect its solvadochromic properties. Next slide. And lastly, we also made an analog of berberine, a library of berberine analogs where we did Grignard additions to C8. And so essentially, you can see like the R group at C8, we were able to put both methyl, ethyl, phenyl, allyl, and tert-butyl groups on that carbon 8, and we wanted to see how this would impact its antimicrobial properties. Next slide. And so to assay those, these are the Kirby Bauer assays that we ran to assay the antimicrobial properties of the Grignard analogs. And we actually published a paper where we analyzed the antimicrobial properties of the methyl analog specifically, but none of the rest of them. Next. We also plan to do MTT assays on these where we basically assay their anti-cancer properties using a colorimetric reaction of MTT. All right. The next facet of this presentation is basically the Monastrol project, where we use Benchtop NMR to monitor the Biginelli cyclocondensation and produce insight into how exactly it works and what reactive intermediates are part of it. So multi-component reactions are really useful in synthesis as they're able to generate synthetic complexity in just one step. Next. And so they're able to generate a very vast library of chemical entities in this manner. And so that ties into Monastrol, which is a mitosis inhibitor. It was found in the Mitchison lab at Harvard. And essentially what Monastrol does is it's a mitosis inhibitor. So it stops mitosis at the metaphase phase, which means that it can be an antiproliferative agent. Next slide. And so the way it works is it's a kinesin EG5 inhibitor, which allows it to be a cell cycle regulator. Next slide. And so what we wanted to do was we wanted to generate a fluorinated analog of Monastrol. This is because while Monastrol is, it's been found to be an anti-cancer agent, it's not necessarily incredibly effective. And so we thought that by adding this fluorine group, we would be able to increase its efficacy as fluorine is, bioorthogonal. So it improves the metabolic stability of the compound because fluorine isn't typically metabolized by the body. Also, because of the electronegative properties of fluorine, we also realized that this would impact the reaction mechanism of the Biginelli cyclocondensation by switching the polarizability of the compounds. And so as you can see here, adding on this trifluoromethyl group has been really utilized in many different antiproliferative agents that have been FDA approved. I think around 18% of FDA approved drugs were fluorinated. And so we wanted to see if we could do the same for minastrol. All right. And so, like I said earlier, we also wanted to see how this would interact with the Biginelli cyclocondensation as a whole. Next slide. And so in order to do that, we used a trifluoromethyl methylated acetylacetate. So basically our ketoester that has a trifluoromethyl group on it. And we were able to use that in order to incorporate the fluorine onto the minastrol scaffold, as you can see there. Next, yeah. And so while we were synthesizing this product, we were unable to get our, our expected product. As you can see up top, our expected product is basically just one with like a trifluoromethyl group, but instead we found that there was actually an extra hydroxy there. So it was like the hydrated version. And so we weren't really sure why we observed this product, and that only inspired our curiosity into the different reaction mechanisms. And so there are the three possible routes of the Biginelli that have been reported in literature. The first is the Iminium route, where we have the thiourea and the benzaldehyde react first. And then this then reacts with the ketoester to form the final hexahydropyrimidine. The second route is the enamine route, where we have the thiourea and the ketoester react first. And then you see that this forms the imine, which tautomerizes. It reacts with the benzaldehyde. And then finally, if you notice this step right here, where we have this hydroxy elimination before this final cyclization to the hexahydropyrimidine. And then the last route is the Knoevenagel route, where we see that the benzaldehyde and the ketoester react first. This undergoes an aldol condensation, which then undergoes a beta hydroxy elimination, and then it reacts with the thiourea and an azomycyl, and then it finally undergoes a final cyclization to get the hexahydropyrimidine. And so we wanted to monitor this reaction, but you can't use like a microscope or like even a scanning electron microscope. Atoms are much, much smaller than those can detect. And so in order to assay things on a molecular level, we had to use a very small instrument, basically the NMR. This is a benchtop NMR. This is the one we have in lab. And this one's able to actually monitor reactions at a molecular level. Next slide. And so to determine the mechanism, we used benchtop NMR in order to figure out which one of these reaction mechanisms was operative. And so first we ran experiments where we excluded one of the reagents. So as you noticed earlier in each of the reaction pathways, each of the pathways start out with 2 of the reagents rather than all 3. And so we excluded the necessary reagent in order to force conversion to one intermediate. And then we integrated that to find how much of the intermediate we had. And then another thing we did was we developed a Hammett linear free energy relationship. So essentially we created this library of minastrol analogs, these hexahydropyrimidines. They basically just have different aryl substitutions. So we wanted to see how electron withdrawing character at the benzylic carbon would impact the reaction rate of the Biginelli. And so we saw that here the NMR kinetics, you can see that we just integrated concentration over time to develop this relationship of like product versus time in order to find the reaction rates. Thanks. And then here's just the Hammett. So basically it just talks about electron withdrawing character. Thanks. And so by using the different Hammett values of the different aryl substituents, we were able to graph that against the initial reaction rate, which we got from the QNMR earlier. And then we were able to develop these relationships for both 3 substituted and 4 substituted aryl substituents. And so we found that for both of those, we had like this negative relationship. So as Hammett value increased, the reaction rate actually decreased. So that indicated to us that there was some kind of loss of charge as like the electron withdrawing character increased, the reaction rate decreased. And so that ties back to our conjecture about the mechanistic route as well as the rate determining step. So because they're both downward, and because earlier in our exclusion experiments, we found that enamine route develop the most amount of intermediate. We believe that the enamine route is operative, and we also believe that the second to last step, so the hydroxy elimination before the final cyclization, we believe that that's the operative or the rate determining step because it has a loss of positive charge. And so to finally corroborate that, we used in silico modeling. So basically what we did was we used DFT, where we calculated the energy of each of the different intermediates in each of the reaction pathways. So the imine is featured in green, the enamine is in red, and the Knoevenagel is in black. And we did that for 7 different aryl substituents. And as you can see here, for every single one of the pathways, the enamine pathway starts with the lowest energy intermediate. And we can also see that there is a sharp increase in energy at the second to last step before this final decrease in energy at the cyclization, which does correspond with our Hammett observed results. Cool. And then moving on, so now we're going to be talking about our antiviral approaches. So the first is we developed this peptidomimetic small molecule inhibitor of SARS-CoV-2's cathepsin L. Next slide. So basically, this is just the mechanism of action of cathepsin L. But basically, this cathepsin L, it's a protein that promotes replication within a cell of SARS-CoV-2. And so what we wanted to target here was the active site of cathepsin L. It has the cysteine 25. Next slide. And so we wanted to develop this inhibitor. It kind of mimics a peptide. And we has like 2 different facets. So there's an epoxide trap, which actually traps the cysteine 25 that I talked about earlier. And then there's that which recruits it to the binding site. Next slide. And so this is just the proposed synthesis of inhibitors. I don't think we've followed through with this too much, but essentially can you go back? Yeah. So how our inhibitor works is it basically does a Michael addition with the cysteine 25. It falls into this epoxide trap, and then it creates a covalent bond with the cysteine, with the thiol on the cysteine. And so this is important because it means that since it's a covalent bond, this is irreversible, which is really, really important for making sure that the virus does not replicate. So what we did here is we used a lot of in silico work. So we used density functional theory as well as molecular docking. First, we used density functional theory to basically optimize our structures and mimic how they would be in the environment. And then we used molecular docking to see how strong these interactions would be with cathepsin L. Next slide. And so we did 2 versions of the inhibitor. We did one where the R group was forward-facing and one where the R group was backward-facing. And as you can see here, we found that R groups with like Arginine as its R group were really had really good binding affinity. Next slide. And so we also looked at the binding poses with the native peptide, and you can see here that one of their best analogs, lysine, it had very interesting binding. It had really interesting binding poses with cathepsin L, specifically with the back and front epoxides. The epoxide group is just very positioned very differently. And so I think like one important facet of this project was the fact that it was completely virtual. We did this at the start of the COVID pandemic, and so we were able to do this work even though we were forced to be virtual. And so that allowed us to discover these inhibitors. And while our in silico results weren't the greatest and we decided not to continue on with synthesizing them, this did provide valuable insight, and it did allow us to understand like how exactly such an inhibitor would work. And it did inspire, I guess, our next project, which is the Carmifer project, where we make 5-fluorouracil small inhibitors. Essentially, Carmifer is a prodrug of 5-fluorouracil. It's a potential inhibitor of the SARS-CoV-2 main protease. So this is a different protein target. It was initially developed as a acid ceramidase inhibitor, but then it was repurposed in a drug screening for main protease. And so we wanted to develop analogs. So the first thing we did was similar to the last project, we did molecular docking. Again, it was this workflow of density functional theory to molecular docking to develop libraries of analogs that would be suitable to inhibit the main protease. We also did homology modeling to see how it would impact its binding and molecular dynamics. So on the left side, it's all just in silico work. And then we also did a lot of optimization We did a lot of optimization of the synthesis of Carmifer using Benchtop NMR. We actually synthesized it, and then I think we're going into biological testing now. So next slide. So for inhibitors, essentially what we had is we had different chains where we had either an alkyl side chain, an amino acid side chain, or a benzene side chain. And so for the alkyl side chain, we wanted to see whether or not having this really hydrophobic property would impact it. Amino acid side chain, we wanted to see if mimicking the native peptide would improve its binding affinity. And then benzene side chain was kind of just to see how the size would matter. And so next slide. And this is what our molecular docking. So for this one, we also targeted one of the cysteine. I think it was cysteine 145, and this is main protease. Next slide. And we also used homology modeling, which allowed us to mutate the SARS-CoV-2 main protease to see how our analogs work against mutated versions of COVID. Next slide. And we also did molecular dynamics, which just basically gives us a more detailed understanding of how exactly the inhibitor works in the native environment, as you can see. Next slide. And so we eventually found that the binding affinity of just Carmifer itself, it was -6 kcals per mole. So this is not that great of an inhibitor. So again, the aliphatic tail forms hydrophobic interactions, which is why we assayed the rest of the alkyl analogs. Next step, or next slide. Also, we found that for the amino acid analogs, we found that the tryptophan analog was most helpful, where it had the best binding affinity. And then for the aliphatic analogs, we just increased the tail length of the aliphatic group, and we found that those with longer tail lengths actually had greater binding affinities. Next step, or next slide. Finally, for the benzyl analogs, I think we observed that these ones were not as did not perform as well as the other analogs. It didn't enter the S2 subsite as much. So basically it's like a little hydrophobic group in the main protease. And so what we ended up doing is we continued with the synthesis of these Carmifer analogs, where we had, groups with alkyl chains, but we also had different lengths of aliphatic chains, and we also had different substituents on the bottom. Next. And as you can see, there's like a fluorine handle on it, which made it really helpful for the synthesis of Carmifer and its analogs to be tracked by Benchtop NMR, which also allowed us to develop an understanding of what the reaction rate was like under different conditions, and also just allow us to optimize the reaction in general. Next. Yeah, so thank you so much. I know that was a very, very long presentation, but thank you so much for everybody for listening, and also Edward and the rest of ASDRP, Mr. Downing, for keeping up with my server. All of these things that I presented probably crashed his server many times, and I'm so grateful to have the opportunity to engage in such research. And yeah, thank you so much. Any questions?
Yeah, well done. And you know what?
Thank
As
you.
usual, I have tons of questions, but I'm going to give up. I'm not going to ask any of those questions. And next time when Edward's team has a colloquia, I'm going to revise their presentation. That is for real, Edward. Like, for real. Like, your kiddos cannot go like 70 slides. Like, this is violation of colloquia.
22 minutes. 22 minutes. I'll try and go.
It's fast.
Okay, well done, Sarah. I have a question, but trust me, I'm not going to ask you. So I'm going to let you pass.
Okay, any questions from the audience for Sarah?
A bunch of people walked into the library like in the middle, and they just like left.
You scared them.
Yeah.
Okay, questions. Okay, somebody messaged me instead, but they said very impressive. Okay, so you have a hand up in the audience.
Harriet or Ashwin first? Or I guess I'll just answer Ashwin's question first. Yeah, so during the pandemic, we didn't have access to the lab for like I think until May, and then we were able to go back in person wearing masks and face shields. So we did eventually have access to, things required for synthesis. But in the meantime, we just modeled them on our laptops. So yeah.
Excuse me, laptops?
Well, no, it was on our laptops then, because we didn't have the server then. So I would literally run DFT at my school. I would ask my teachers if I could keep my laptop open in the back of their class and plugged into a power source so that it wouldn't shut down in the middle of class. Yeah.
We built the cluster during the pandemic.
Yeah, but I
We?
didn't.
You live things.
Harriet?
So, Sarah, just one more thing. When you're adding fluorine to any small molecule, for sake of drug discovery, there's always advantage and disadvantage. So it will be good also that you emphasize on the disadvantage of adding the fluorine, which is the insolubility, which is so there is a limited solubility, I think, right, Edward, for fluorine?
I think there's greater stability or greater metabolic stability. I don't know about in the file.
And it has the pharmacokinetic limitation because it's vitro, right? You're not testing on a human. So so it might sound very good to add those to the small molecule, but not always. Like when it comes to human trial, it's kind of a drug that can be, administered, right? So there are always disadvantage of fluorine when it comes to human trial, right? So maybe you want to emphasize that there's not always like by adding any fluorine to any small molecules, yay, we got a drug, right? So anyway, so I think you have two more questions. Is that okay if my team goes, because I have to go fast?
I need to ask a question.
Oh, hi.
Hello. So Sarah, you said that at one point you were finding a unusual group that was being added at 0.8 on the chain, and that you were doing some experiments to determine how it got there.
I don't know if I understand your question. Sorry.
Okay. Back, if I saw your slides way back in the beginning,
Larry, do you remember which slide?
I'll recognize it when I see it. Keep going.
Oh, it might be.
Whoa, back up. Oh, wait a minute. Keep going. When you were adding the fluorine, when you first started adding the fluorine.
Mm-hmm. Oh, are you talking about the dehydration? How we observed the hydroxy group?
Yes.
Yeah. So the hydroxy group, essentially, it's just here. Basically, what we expected was that we expected just to see like a alkene there. As you can see between the trifluoromethyl group and the carbonyl. But instead, we didn't observe that. We observed that hydroxy there, and it wasn't necessarily like a bad thing. It's just that we didn't get our expected product, and instead that to get the expected product, we have to actually reflux with triflic acid 3 equivalences. So it's just kind of not fun to do. But basically, it just provides questions into why the mechanism varies when you add this fluorine group versus when you don't add this fluorine group. I don't know if that answers your question.
So you said that there were 3 reagents that you used to add the fluorine group, and you were concerned about which one began the.
We were concerned. It was the ketoester. So we use 4,4,4-trifluoroethyl acetoacetate. And so since we have that, this trifluoromethyl group on the acetoacetate, that kind of switches the polarizability. And so it makes it more electronegative. And so basically we think that would switch the mechanism from the imine to the enamine group, or enamine route, sorry.
Yes, thank you. Okay.
Yeah.
Sahar, you can go ahead.
So in the interest of time, if you have questions, email Sarah, sarah.sue@asdrp.org.